Cloning by Kristina Moreno. So, what is cloning? There's three different types. The first one is gene cloning, which is also known as DNA cloning, which creates copies of genes or segments of DNA. There's reproductive cloning, which makes duplicates of whole animals, so that's what we associate cloning to be. There's therapeutic cloning, and it creates embryonic stem cells, which are used to create tissue that can repair or replace damaged tissue. So that would help when talking about diseases. How is cloning useful? We can clone cells for untreatable diseases, which are like heart problems, diabetes, spinal injuries. It could give children to those who are unable to conceive, and you would be able to avoid passing on diseases to your children because you're essentially picking what you want for your child when going through with this process. What is the status of cloning? In 1979, researchers produced the first genetically identical mice. Shortly after, they produced the first genetically identical cows, and then sheep, and then chicken. In 1996, they succeeded in cloning the first mammal from a mature somatic cell taken from an adult animal. After 276 attempts, they finally produced a lamb, and her name was Dolly, although claimed it has not been performed on humans. The controversy of cloning. Is it ethical? It's not the traditional way to have children. A lot are going to argue. The process is wrong because you're essentially making a baby inside of a test tube. It could give false hope to those who have sick relatives or children, and it can be unsafe, especially in humans. It causes physical and internal abnormalities. The few clones that do survive are very unhealthy. Abnormal placentas or abnormally large fetuses can kill a surrogate mother, and most clone animals die during gestation, so it's not safe at all. And these are my sources.